It's great to have been invited. Thanks to the organisers for giving me the chance to come to this, what I'm sure is going to be an absolutely wonderful meeting. I'm also very happy to be the first speaker. Get it out of the way. So just going back to the first slide here, apologies for the slight pun in my title, but I run a group based in Oxford, and for this meeting, certainly we're going to be at the very sort of applied end of things. So my group works on developing model-based inference under models, especially the coalescent, to understand a variety of different forces that I'm sure we're going to hear lots about at this meeting that affect us. For example, fine-scale population structure, admixture migrations. A key interest in my group has been studying recombination rates, natural selection. I'm going to touch on a few of those in this talk. So what I thought I'd talk about, given the sort of general area of this meeting, is some work that I've done with my group and collaborators, mostly in the UK, on developing methods to take genetic variation data. So, for example, if we sequenced all the people in this room, and to use that genetic variation data to build genealogies across the whole genome. And what I'll talk a bit about is the idea that although those methods, because the problem's so complex, build genealogies under a very simple and somewhat naive model, hidden within them, that's the pun of the title, is a lot of richness that's captured by departures from those simple models that we can also in itself model to learn about the things we care about. So it should become clear what really I mean by that as I go. So this is what we're going to be focusing a lot, at least in this talk, is the idea that if we take a lot of people, at each position in the genome, we're related until a common ancestor by a genealogical tree. Mutations happen in those ancestors, and importantly, those trees change as we move along the genome. And for humans, these genealogies go a long way back in time, at least 3 million years. I'll show you something even older than 3 million years, so we can learn about the distant past. Okay, so as I'm the first speaker, and with apologies, I thought I would just start with a very simple model, perhaps the simplest sensible model of genealogies, and discuss that a bit, particularly because, as I say, it's the model that our method, which is going to be called relate, performs inference under. As I say, nice gentle introduction. So let's think of a single position in the genome. As you know, as I say, if I take the people in this room, I get the pointer to cooperate, maybe I'll point at the screen. If I look at that position in the genome, then as we go back in time, we can follow our ancestors, and eventually we reach a single common ancestor. And the population size may vary back in time as we do that. Kingman in 1982 proved that under a variety of related models, we can think of that process as being described by the coalescent. And although there are sort of diverse models in the details, they all share a single property. The key property that we need is that in the previous generation, we imagine that our parents, our ancestors, are chosen uniformly at random from all the possibilities. So there are NT possible choices. They're all equally likely. Now, if that's the case, then if we take a sort of pair of ancestral lineages, say these two over here, in each previous generation, the first lineage will choose an ancestor. The second one then chooses the same ancestor with that probability 1 over n of t. And that implies that under sort of mild modeling conditions, and provided n of t is large, we can approximate this whole thing by saying, well, rather than in discrete generations, for example, coalescence happens at some rate, 1 over nt per pair. So while we have j edges in this tree, say across there j choose 2 possible pairs, and so the rate of coalescence becomes j choose 2 over nt. And this fully specifies the model. For example, we can simulate data under this process. Something I'm going to come back to briefly later, I'll just highlight. Because of symmetry, if we sort of ask forward in time, which of these lineages is most likely to split? Well, they're all equally likely to split, so it's just 1 over the number of edges, that probability. And finally, so we can already simulate genealogies under that model. If we want to get data, we need mutations. or we can just introduce a Poisson process, a constant rate of mutations happening, and drop them on the edges, and that generates diversity patterns. Okay, so I'm sure many of you are going to discuss sort of extensions and more complicated versions of this model, but actually, in terms of inference, we've given the mutation patterns in these samples. Inference under this model is still absolutely the best we can do. So all the leading methods just attempt, actually often approximate inferring histories under this model. And the reason for that is a sort of classical problem in statistics. It's very easy to simulate data under this model, but if you take the data, the reverse problem of inferring the genealogies is much harder, essentially because there's a huge space of possible trees that could give rise to the data we see. And I'm going to talk about our method a bit, relate, or really extensions of this method to do that. Okay. All right, so I mentioned also that trees change along the genome. So again, as I'm sort of introducing things, that happens because of the process of recombination. And so the simplest way we can think about recombination is, suppose I think about one of my chromosomes, say my maternal chromosome 1. I obviously inherit that from my mother, who has 2 copies of that chromosome, the black one and the red one, from my grandparents on her side. Usually she doesn't just pass on one or other of these to me. Instead, I inherit this kind of carefully constructed mosaic of her 2 copies, where there's a very precise switch from one to the other. That's called a recombination site. Now, I love to study recombination. It's a fascinating process. But for the purposes of this talk, we have to think about what this does backwards in time. And so if you now think about this as a stretch of DNA and this is one of our ancestors, this process can also happen in our ancestors as well. And we can model that. So the idea is, suppose that I have a genealogy at some position in the genome like this, and then the ancestor of this lineage D may undergo one of these recombination events in this region. So we can just think about what happened. So now thinking backward in time, the arrow of time has changed to look backwards. To the left of that breakpoint, the ancestor is this individual, and we can follow their ancestral lineage, the black one. But to the right, we have a different ancestor from a different individual. And so we have a distinct lineage, and because they're distinct, they do their own thing. They can coalesce separately, and this is what results. And so as you can see, the history, rather than being a tree in this region, becomes a graph where, if you like, just like investments, the number of ancestors back in time can fall as well as rise. So we get increased number of ancestors when we have these recombination events. Now, the result of that is we end up with a sort of sequence of trees along the genome. So to the left, we follow this way, and we get this tree where I started. But if we sort of take this position here to the right, the ancestral lineage is this one. So sequence D coalesces with sequence E, and we get this tree. As we build up recombination events along the genome, the trees change more and more until they become essentially uncorrelated. Now, it's really trivial in some sense to add recombination to the model. We just assume it happens with constant probability, so we get a constant rate of these events on the edges. The problem comes actually when we try and do modeling calculations. It makes things rather messy. But in principle, it's easy to simulate data under this model. And this process was, of course, discovered shortly after the coalescence itself. Okay, so what I'm going to talk about a bit is not so much how we generate genealogies from data, but kind of what we can do with them. And the basic idea is, we in this room collectively might be interested in all sorts of things, like this, covered by this list: natural selection, maybe population structure, migrations, demographic history, whatever you want. All of those, in general, tend to impact our ancestry, if we're doing population genetics. And so what they do is they impact this distribution of correlated trees along the genome. They make the model more complicated in a sense. And in turn, those genealogies define the variation patterns we might see. So if I gather a data set, maybe it looks like this. So notice then that sort of these forces only really impact data we see through the genealogies. So an attractive thing to do in terms of capturing information would be to try and infer these trees from the data and then sort of read off details of these processes. That's what we should do. It would capture all the information in the data. The problem is, as you can perhaps see just by this little cartoon, it's non-trivial to go from here to here. More than non-trivial. It's extremely challenging, and the data sets are massive these days for humans. So most approaches haven't usually done that in the past. Instead, what we do is we take this data set, we're informed by the ideas of the genealogy, we tend to construct summary statistics, informative statistics, in some sense integrate out the trees and then learn about these processes. So the question is, could we be more direct? And until quite recently, that's been very, very challenging. So I should say that methods have been worked on for inferring these genealogies from data since at least the early 90s. Joe Felsenstein, Bob Griffith, Simon Tavray, other groups worked on it, but they didn't scale very well. So they're very slow and could only do the most basic models. But in the last several years, in particular with this class of methods here, there have been some advances where we can at least infer histories under approximations to these models. And the key thing is, they were all published pretty recently. They're from different groups. For schizophrenic reasons, two of these groups, including mine, working in Oxford, solve a problem separately and publish methods. But the key thing about all these methods, they have strengths and weaknesses, but they can all scale to realistic data sets with thousands of individuals across the whole genome. So it opens up that possibility of great power in our inference. So I'm going to talk about applications of this method relate without talking so much about the method today because it's already published. Because I'm talking about applications, the key ideas are largely method agnostic. So I'm only picking relate because it's from our group, really. Has some properties that are helpful, but we could often use other methods. Okay, so a little overview of this method. So I'll be very quick with this slide. So yeah, here are the papers. You could read them if you're interested. But the key thing to know, this method appears to work well enough in practice that hundreds of different studies have used it in a variety of species. So here's what it will produce. This is one genealogical tree at one position in the genome for a human sample, a gene called lactase, which is thought to be under natural selection. I'll talk a bit about selection in a minute, so I won't talk much about this. So you get this nice structure. By looking at these trees genome-wide, you can investigate a variety of phenomena. So, for example, estimate the population size in the past, look at natural selection, or understand the changes in the rate of mutation evolution through time, all using the same collection of trees. So you get self-consistent inferences. So I'll talk a bit more about some of these applications in a modeling sense as we go. So a few things that it might be helpful to know. We estimate the population size through time, as you can see here, but otherwise we use that very naive model that I introduced in the first slide or second slide or whatever. It's pretty fast, but it tries hard to be robust to natural errors in the data set. We can build ancient DNA samples into the same trees as modern samples. There are already quite a lot of add-on tools. I'll talk about some more in development. Once we built these trees, we can map mutations onto the edges and see where they happen on the tree, and we estimate branch lengths and, as I said, the population size. So what I won't tell you about is how we build trees using relate, but believe me, it's very clever. So actually, it's not like it's built from a series of simple principles, but with the key focus being we have to make it fast enough. So it's not just got to be accurate, but it has to be fast enough to work. So here's a data set I will talk a bit about where we've applied relate. You see the kind of scale. This is for 5,000 haplotypes from 26 populations. So it's a worldwide sample, but we built the trees for all of these individuals together. So we assume them to be from this panmictic population. Obviously, they clearly aren't, and we might think about that. At 71 million SNPs, most of which we can map onto the tree edges that we produce. We assume an infinite sites-like model. And this took about 4 days on a cluster to do so. It's pretty computationally intensive, but it's certainly doable. So key ideas I'm going to extend a bit more. So as I've said, the idea is we want to analyze lots of phenomena relating to population genetics by using the same set of trees genome-wide. But what I want to sort of extemporate a bit more about is this idea here. So most interesting things that we may care about turn out to relate to departures from the model we built the trees under. So if we wanted to study population structure, that's a departure from panmixia. And so I'll talk about the idea that actually it would turn out that even though we built trees under one model, they capture features of those processes, and we can model the way they capture features of those processes as well. And so here's a simple example that's already published at the same time as Relate of how we can do this, how we can find departures. And this is the idea of saying, well, suppose that we want to know whether there's natural selection, specifically positive selection, acting on some mutation. So the mutation happens here on our tree. And the basic idea is, suppose that this mutation is selected. So what do we expect to happen? Its carriers will have more children. And so what we expect to happen is that the carriers under this mutation will have more children, and therefore there'll be more descendants of that lineage than the other lineages in the tree at the same time. So to capture that, what we do is we sort of wait until the lineage first splits for technical reasons, so it has 2 descendants here. We count the number of lineages that are present when it does that, and then we also count how many children in our sample of N haplotypes at the bottom of a tree here this mutation gives rise to, so that's some number Fn. And now we go back to that fact that I highlighted before where, because all lineages are equally likely to split, the number of descendants of any lineage, or the joint distribution of all the lineages rather, has a uniform distribution on all possible partitions of our sample of N into these K lineages. And so we can write down the probability that we have Fn children. So here it is, it's just a number of partitions where we have Fn descendants of these 2 lineages. That's this product on the numerator divided by the total number of partitions of N things into K groups. Key thing is this is analytic, we write it down. So then we can get evidence of selection by saying, well, how likely is it we see this many or more children? So we just add up these probabilities. So as I say, it's a simple example. It will get a bit worse as we go. Now, that's a sort of simple, it's the first idea we had actually. But it turns out that if we simulate data, I won't show sims here, but it's very well calibrated. It's got a uniform distribution. And it already turns out to be more powerful than the other existing approaches at that time that don't use genealogies. So there are methods like IHS and EHH and many other groups have made approaches. All those we tested were less powerful than this simple idea, and it's been improved upon since, including with machine learning type approaches. And so that's one example of how by using trees we can benefit things. So here's a place in the genome that this test identifies. So this is the tree, a mutation, a non-synonymous mutation in a gene called EDAR. And this mutation itself has been shown to be associated with a variety of phenotypes like thicker, straighter scalp hair in humans. And you can see that the mutation is only really present in East Asians, and the carriers certainly do seem to sort of dominate individuals now, despite this mutation having arisen and spread very recently. If we look genome-wide, this is one of the top signals in Chinese Han population as shown here, where we see others. Here, the UK signals, we see different things, including that lactase gene. And just as an aside, there aren't very many very significant things in that plot. We think that's a feature, not a bug, because actually, as you probably know, human traits tend to be very polygenic. And that makes selection very weak on individual mutations. And if we actually look on traits, we do see widespread evidence of selection on human traits. For example, height or directional sex selection towards lighter hair color in Europeans and so on. All right, so I'll stop with selection. You can ask me more about it later and move on to a different question. What I want to sort of illustrate is that this happens again and again. So the second kind of departure And this, by the way, this work was all done by Anna Ignatieva and some collaborators. While she's a postdoc in my lab. She's now PI, Glasgow. And it asks a different kind of question. So suppose, like me, you're interested in recombination itself. So as I said to you, our trees are built under a simple model that says all the lineages have an equal probability rho over 2 rate of recombining. So you might wonder whether, like selection, which causes some lineages to split faster than you expect, lineages might have different recombination rates to each other. And we got interested in that, motivated by the idea that this should happen. So a variety of processes might be responsible. So we suspect that genes expressed during meiosis have suppressed recombination. So EQTLs for those genes might cause different lineages to have different recombination rates. But the one I want to focus on is the idea that we expect inversions, so regions of our genome where marker orders get flipped to suppress recombinations. So I'll just show you briefly why that is. So it's from biology, basically. It's almost known. So the idea is, as shown here, suppose we have a region of the genome where the marker order is A to F of these positions along the genome, and they become inverted in some individuals so that we go A, E, D, C, B, F, as you can see here. Now, what we can imagine is some individuals have this inverted form, others are a wild type, so some individuals become heterozygous for the inversion in their 2 chromosomal copies. And what you can then say is what happens during meiosis. So when you try and, when you have recombination during meiosis, the chromosomes pair, and in this region, something weird has to happen. So we don't need to know the details, but you can see they form a sort of strange circle where they try and arrange themselves to align. Now, if recombination then initiates within this region, say here the DNA gets cut, it turns out that the resulting products tend to either have no centromere, as shown here, that tends to be inviable, or 2 centromeres. Either of those is not a good thing, and so you just don't see those in offspring. They're usually going to be lethal. And so instead you tend to pass on non-recombined forms, either just the wild type or the inverted form, intact to your offspring. So throughout the region of the inversion, we would expect recombination to be suppressed in heterozygotes for the region. All right, so if you swallow that, then the idea is, we can just basically think of it simply as saying that recombination is banned in the region of the inversion, so we don't see these recombinant alleles. Okay, so now what we have to think about is what that means in our ancestral recombination graph, or ARGs. So the start point is to say, well, this inversion had to happen in some ancestor. That's what the lightning shows. So let's suppose the inversion happened in this ancestor and then starts to spread. And this is the tree we have at some position in the inversion. Firstly, non-carriers of the inversion can have recombination between each other. So one non-carrier, this one, and this one can easily, or the descendants can easily recombine to form individuals today. So this is fine. Carriers of the inversion also can recombine with each other. So this carrier and this carrier can recombine and produce this offspring. But what we cannot see under this model, so that's what the X means here, is recombination events between carriers here and non-carriers here, wild type. So for this event to happen in the past, this individual here, or at least their immediate ancestor, would have to be heterozygous for the inversion and then recombine, and that can't happen. All right, so what we expect to happen is the ARG doesn't contain events like this, where if you like a carrier here recombines with a non-carrier. So the effect of that is this clade is kind of isolated from recombination with other parts of the genealogy, and so we expect this clade to remain intact for the whole region of the inversion, if that makes sense. And that obviously departs from our model where all lineages can recombine in the same way. And so what we could ask is, can we detect inversions by looking for clades like this one, that persists for a long region in the genome, kind of not broken up by recombination. And this would actually be useful because inversions are notoriously hard to identify in real data by other means, essentially because with short reads, you can't map across the inversion breakpoints. Now, what we then have to do is to say, well, how do we define odd? So here's an example region that we might think is odd. So these are real trees now in chromosome 17. So at this position, we have this red clade here, and you can see all the carriers in blue at the bottom. And if we go far enough away to the left, that clade gets broken up, all those carriers are in some different tree, or far enough to the right. But actually, even if we go 700,000 bases away, you can see that clade is completely intact, whilst all the rest of the genealogy looks like it's changed greatly. And so that might be the kind of signal we need. So perhaps this is longer than expected, but actually we need a test to ascertain whether this is really the case or not. And so we can't actually, the problem with the ARG is it's really hard to do exact calculations. We can't actually do this calculation under the ARG. And so instead we've used an approximation called the SMC prime model that I'll just briefly talk about. So SMC prime was published back in 2006, but since then it's been quite widely used as a subtle approximation to the ARG model, or the full coalescent with recombination, because it's a highly accurate approximation. In fact, at first glance, you might think it was correct. It's not quite correct. And it's certainly much more tractable for calculations because it models genealogies as changing as a Markov process along the genome. So the idea of a Markov process here is if at some position, this one here, I know the tree, I can get the tree at the next position just by conditioning on what I have at this site and then evolve things as I move along the genome. And so what we can do is define rho to be the recombination rate. We're allowing this to vary along the genome so we can have recombination hotspots. And at this tree, we sum up the branch edges to give a total branch length L of t. And the idea is that's the opportunity for recombination. So the distance until we get the next recombination event is just a Poisson process with rate rho Lt over 2, or drawn from a Poisson process. So at some point, we have a recombination that's shown by this red arrow that happens to any part of the tree chosen uniformly at random. Once we've chosen where the recombination happens, we allow this edge to coalesce with the others as shown. So it's its own lineage. This one coalesces here. And so now if I look to the right of this position, now sequence A and B will coalesce with sequence C and D at this time here. And so the next tree to the right becomes this fellow here. So we evolve the genealogy under this model. And then we just repeat the process. So we again calculate the edge length of this tree, have an exponential weighting distance, have a new recombination event, and that results in a change here. So notice the TM-RCAs, the whole tree can evolve as we move. As I say, I can talk later about it with people, but this is in fact not exactly the right model. It's an approximation to it, but it's a close approximation to it. Now, under this model, we actually can calculate what we need. So we need to think about how long, for example, does this clade persist as we move along the genome. And so now I have a couple of slides with a few formulas, but I won't show you all the formulas, I promise. So to answer that question, suppose we have a tree here, and we're looking at this clade G. So these blue edges here, and we want to know what's the distribution of how long that persists along the genome. So we notice firstly that G can only break up when we have a recombination event. So we have to think about recombination events, and different things can happen. So firstly, we might have a recombination event within the clade G itself, as shown here. So that's what the X shows. That recombination event then coalesces somewhere back into the genealogy. If it coalesces here, notice it changes things around within the clade, but it doesn't stop the clade from existing. It doesn't break it up. But if instead it coalesces over here, now sequence 0 is more closely related to sequences 3 and 7, so that does break up the clade, Similarly, if the recombination event happens over here, on this edge, then if it coalesces back up here, that doesn't affect G at all, so it maintains. But G is broken up if the coalescence event brings you back inside the region here, because now we have a nucleid member. Okay, so the probability that a recombination event breaks up the clade, it never does if it happens on this edge, is just the probability that it happens within the clade G and then coalesces outside the clade G, or the converse, it happens outside the clade G or coalesces within the clade G. There are other rules that we have to take care of. It can only coalesce further back in time than the recombination itself happened. And that complicates the calculation, but in principle, we can do this. This is saying what happens if there's a recombination event on a certain tree. So it has 4 terms. We can calculate each one. And I'm tempted to go through each term, but maybe we'll be here all day. So just to say, it's messy. Obviously, there are terms in here that I would need to define, Q tilde 1 and Q tilde 4, before you could understand it. But the key thing is it's tractable, it's doable, and it's actually computationally quite fast to do. We just have to keep track of a bunch of things. So suppose we have calculated each term, then now G just breaks up again as a time inhomogeneous Poisson process. So each time on a tree T we have a recombination event, it has this probability of breaking up a clade, and the rate of a recombination on the whole tree, as we've said already, under SMC prime is this. We have the span of G just being the waiting time in that inhomogeneous Poisson process. Notice the rate changes along the genome because the trees change along the genome. That's why it's not homogeneous. And we can turn that into a test for whether a clade lasts longer than it should. So the idea is, suppose we have a tree T, we look at some clade G, and we get its span D. We can integrate this rate and get a quantity Q, and then the probability that we see survival of our clade for at least as long as D becomes a p-value, and that's just e to the minus Q. All right. We have another test which, rather than sort of using the distance itself, says, well, how many recombination events in a row don't interrupt my clade G? In a similar calculation, we can work out that p-value. And we use both tests in practice because they have different false positives. All right. And I've brushed over a variety of things, which I will continue to brush over. But we do make adjustments, as we'll see they're useful, to allow for various errors, basically, in the trees we built, because the trees we built are not perfect. And we also approximate this inhomogeneous process as homogeneous, because these terms don't turn out to vary much along the DNA. Okay, so if we take a little breather, and we can just sort of relax and look at some simulation results for a minute. So one thing we might say is, we've got this magic wand for saying whether, there may be an inversion. Does it actually work? So we simulated args under the null. So we really have just got a homogeneous recombination event. So this simulation is for 100 sequences along chromosome 21 rather. And they happen to have a constant population size, but we can do a variable population size or a different sample size. The results are much the same. And then the key thing to ask is, under the null, are our p-values uniformly distributed as they should be? So we make a QQ plot showing the quantiles under the null, they're just uniform. Against those we observe by actually constructing this statistic on the previous slide. And the hope is we want to lie along this diagonal. You can see not all, already see, I think, not all the lines lie along the diagonal. So firstly, the one that does, the light blue line here, square along the diagonal, that's p-values using the real args, but still using that SMC prime approximation to get the p-values. So that approximation itself works very well. Okay, so we also suggest that that horrible calculation was perhaps done correctly, because if it had gone wrong, I don't think the p-values would be well calibrated. So next, I have a variety of other lines, and they correspond to using some of these modern methods for building genealogies. And what you can see for all methods in these colors here is that they depart strongly from null expectations. In particular, they show too many long segments. And so before you go away thinking that this means all the methods are useless, bear in mind that this test isn't really fair, because these methods were all designed to be good at one problem, which is building a genealogy at some position in the genome, and all of them, including our own, had to make compromises in terms of correlation structure between trees to do that. And that leads to sort of a conservative approach to changing trees, which means you get too many of these long segments. However, the key thing that makes this useful is, for our method at least, relate, where we can sort of use the nuts and bolts of the method. Those adjustments I showed you on the previous slide, where you do things like only looking at edges if they carry mutations. Apologies, I didn't mean to go there yet. They basically fix things. So this green line after the adjustment now does show a nice distribution of p-values. They really do look uniform even into the tails. And so we went ahead and applied that test to real data. 5 minutes, okay. So here's what we see. So as we move along the DNA, we see a bunch of positions where we see strong evidence of departures from that null model. And so I'll just sort of briefly mention what they say. So we test, this is a test of all the clades for all the trees in the genome, with every point being a test. And for those points that are significant by both test 1 and test 2, we color which population we see things in. And the key thing is we see lots of departures from that simple model of boring recombination in different groups. So perhaps the most significant feature in the genome is the example I highlighted before. That's actually a well-known chromosome inversion, so it really did work for that problem. And again, encouragingly, a variety of the other hits correspond to known inversions from dozens of different studies, probably, that, as you can see here, from all sorts of different time periods, but this one method picks them all up. But perhaps really nicely, many of the hits, as you can see, don't have the red arrows, including this one, which is maybe the second top hit in the genome, and this one, and a couple of others, we think correspond to novel inversions that weren't seen before. And then we're still investigating lots of the other hits where it doesn't seem to be an inversion. We think there may be some other cause. Won't talk about that. And the reason why we think this one and some of the others are likely to be an inversion is if we zoom in on this region of the genome, we can see the inversion seems to be present in many human populations. And it sort of begins and ends here and here. And that corresponds precisely to locations in the genome where we have repeat DNA in an inverted orientation. And that's known to be likely through recombination, again, to mediate the formation of an inversion. So you have this piece of DNA the wrong way round, it can misalign in meiosis, and then the DNA in between gets flipped. And that seems to have generated an inversion in this case. And several other examples look the same. Okay, so I've got 2 minutes left, so I will only tell you the last piece of the story. So I nearly wrote stretch goal on this slide because I knew I wouldn't get to it. So I'll just highlight something else we can do without being able to give you details, but there'll be drinks and things later, hopefully, to do that. So the final thing that we can do is understand admixture. So without going into the kind of words, just being very brief, the idea is when we build genealogies with relate, we assume all lineages are equally likely to coalesce. But actually, if, for example, we take a simulated data set, our genomes are much more interesting than that. So many human populations have things like segments of Neanderthal DNA, as we know. So we can imagine that the genome looks like this, where we have these white segments of Neanderthal DNA. Now, in these regions, in most of the genome, if we take an individual from China, say, it's more likely it has a high coalescence rate of other individuals from China, as shown here. But in these Neanderthal parts of the DNA, it's likely to be more closely related to a Neanderthal. We can detect that from the trees, briefly. And so we have this approach, it's called Ghostbuster, which basically looks at a tree regionally, takes a target sample and says, does it look like this? Does it look like a Chinese sample member, or does it look like a Neanderthal sample member? And as you can see, this can work very well in identifying, without prior knowledge, regions of the genome with Neanderthal ancestry. And the ghost part of it comes that we can actually iterate this and identify groups, even if we haven't sampled them, by properties of the trees themselves. And so I guess as I have no time, I'll just finish with sort of one punchline of doing that, something interesting perhaps. When we again apply this method in practice, we see things that we hadn't known existed before. So for example, we could observe that if we take individuals from sub-Saharan Africa, they unexpectedly, no matter which population we look at, always contain segments of DNA that are not Neanderthal necessarily, but in fact closer to people from outside of Africa. So most of the genome is closely related to other African samples, these red lines here. A couple of % of individuals from Khoisan population, southern Africa, very far away from non-African groups, are more closely related to West Eurasians. And we are certain that this signal is both robust. We see it in all the populations we look at. We see it in ancient DNA samples as well. But as well as being robust, it's very old, between 5 and 20,000 years old. So unexpectedly, people have been crossing the Sahara, perhaps, for tens of thousands of years before, we've even, these events are dating back to probably before the emergence of modern towns. We already see these long-range migrations throughout Africa. And these completely explain something that people had observed before, which is that if we look at populations in Africa, this is a percentage, so they have a very tiny amount of Neanderthal DNA. So although Neanderthals are thought to have mixed with people outside of Africa around 50,000 years ago, so I have maybe 1% or 2% of my DNA from Neanderthals. It turns out that carried on these segments of West Eurasian ancestry was a tiny amount of Neanderthal ancestry back to Africa, and this helps to confirm the idea that this is real, along with strange mutation patterns. I'll leave this slide up because I'm quite understandably having guys standing up. Just to say, we think we can go further back in time and find even deeper events involving deep human splits. So just to, again, I might leave the summary slide up, but the point of this talk, the thing I'd like you to take away is the idea that we can do powerful things with data that are perhaps interesting in terms of modeling. But the key step is, although we must make compromises in how we analyze data, we have to generate histories under simple models. By considering sophisticated models, we can learn about many different factors from the same collection of genealogies. All right, so thanks to all of you for listening, and thanks to all my collaborators as well.
Very nice and friendly talk. Are there questions? We have 2 minutes. So, please.
Yeah.
So, as you... thank you. Hi, thank you for your talk. I was wondering, as you go along the genome and try to detect these change points in the form of the genealogies, if it would seem natural, since you're already really going through the genome, it seems maybe natural and maybe, I don't know, important to be able to introduce in the model the genomic context where you're at. So for instance, if you're trying to find a recombination event, maybe try to model the probability that this occurs based on the sequence itself.
Yes.
Because this is more likely to happen in highly repetitive regions, or even for, I don't know, if you want to detect natural selection,
Yeah, yeah.
You know, like this kind of including this type of context in the modeling.
So I like that question a lot because I can talk about some other part of my work. So in another life, I focused on exactly that question. I was actually the first person who found there was a sequence motif for recombination hotspots in humans. So we absolutely want to do that. So something that we didn't know then and we've learned since is actually that there are different sequence motifs in different species. So every species has recombination hotspots, but they're different in humans and chimps. It also turns out they're different among different people in our species. So it's, you know, recombination hotspots are polymorphic basically, and because of variability in the gene that controls the positions of those hotspots, there are hotspots that are active only in people of African ancestry, say, or European ancestry. And that raises the idea, which we've never been able to do, that we can actually, you know, extend some of these ideas to take the genealogies and look at, sort of going further than you said, not just look at variation in recombination rate along the genome, but also look at variation in recombination rate through time. Because we know if Africans have one recombination map and Europeans have another recombination map, there must have been evolution in the recombination map through time. So perhaps we can look at, you know, Neanderthals, humans, other species, and the hope is we can identify recombination hotspots that don't exist anymore. For example, that have rose and disappeared back in the past, but have still left traces in our genealogy. So thanks for the question.
Well, we are 5 minutes over time, and we should defer our discussions to the ample breaks. And so I suggest that we continue with the next speaker, but before, big applause again.